Hey all, welcome to our lab on photosynthesis. This home activity is going to require some materials, and we'll go through those materials now and how you are going to explore a home activity that will demonstrate one of the byproducts of photosynthesis. The first thing that you're going to need is your digital microscope, and this is so you can take a picture of the final product and share that on our group discussion board. The next thing that you're going to need is a couple small dishes. I made this out of the bottom of a water bottle. I just cut the bottom off and then it left me with this clear dish. You want something that's very low on the edges, preferably clear, and that you can look down into using your digital microscope. You're going to need 2 of these. Here's a bottle. I'm going to make another one. There we go. Now we have 2 little dishes that we can look at our specimens in. The next thing that you're going to need is a couple of leaves. These leaves are spinach leaves. I got these from my fridge. You're welcome to use spinach leaves. It shouldn't be a succulent leaf. It also might be tough if you use something like a grass leaf. So I would stick to something that kind of has this broad leaf look, something that's kind of tender and has a lot of chlorophyll. It's green. You can see that both this leaf, which is a chili pepper plant leaf, and this spinach leaf are both pretty dark. So they have a lot of chlorophyll in them. They look like good candidates for this experiment. This is an example of a leaf that you don't want to use. This is a succulent, so the leaves are thick. You can see that they're a lot thicker than this spinach leaf. And these leaves photosynthesize in a slightly different way, which isn't going to quite work with this experiment. So ideally, try a spinach leaf or something like this chili pepper leaf, or any broad leaf, very tender leaf like this. The next thing that you're going to need is a couple of rocks, 2 small stones. The last thing that you'll need is somewhere where you can hide away this sample dish in the dark. And so I have this little box. I can put the dish in here and hide it away in the dark. So one of these dishes is going to go in the dark. You can see here I have all my supplies, and this is how you're going to set your experiment up. The first thing that you'll do is put some water in both of your dishes. That looks good. The next thing is to trim up these leaves so that they're roughly the same size. You can see that these spinach leaves aren't exactly the same size, so I'm going to cut the bottom off. That looks pretty good. Maybe cut the top off. And now they're roughly the same size, and this is to control for leaf area. Remember that anytime we're doing an experiment, we want all the treatment groups to be as similar as possible, except for the variable that we're testing. In this case, we have these 2 pieces of leaf. They're now cut to the same size. Now I'm going to submerge them, 1 in each of these dishes. I'm also going to flick off any extra bubbles that are on the leaf. So I want it to be as bubble-free as possible when I put it in the dish initially. Once it's in the dish without bubbles, I'm going to place one of these pebbles on top to hold the leaf down on the bottom. I'm also placing these leaves in with the top surface of the leaf down. So the bottom of the leaf is pointing up. Now I have my 2 treatment groups. You can see that they both have a piece of leaf submerged under water, being held down by a pebble. One of these treatment groups is going to go in the dark. I'm going to place that in the dark and that's where it will remain. The other will go in the light, and I'm going to put this underneath a lamp in the house, a bright white light. You could also use a bright window sill, but you want to make sure that there's a lot of light hitting this treatment group. Be careful not to put it too close to a lamp where it could get hot and burn, but any home desk lamp placed a few inches above the water should provide enough light for this piece of plant material to start photosynthesizing. Once you do that, you're going to leave these treatment groups alone for about an hour to 2 hours. After about an hour to 2 hours, check on your treatment leaves. You might notice that there's some differences between the two. Try to capture those differences using your digital microscope. Take a picture and upload those 2 pictures to the discussion board. After that, write a short paragraph, a description of what you think is happening and why you think there's such a difference between those 2 treatment groups. Or maybe you don't have a difference between the 2 treatment groups. Still present the material and explain why you think there isn't a difference between the 2 treatment groups. You'll probably notice that other students have a broad range of experiments. Some of them went really well and some of them don't go so well. That's okay. The idea is that some of you will do a really good job on this and get some really good results. Others will do a good job as well, but maybe their results aren't so good. And we can talk about why that is on the discussion groups. So, prepare your materials, do your home experiment, and I'll see y'all on the discussion board.